Hello, my name is Philip Michaels, and I am a solution architect here at Tetra Science. Today I'm going to talk about how we can accelerate upstream bioprocessing with the Scientific Data Cloud. Let's take a look at a typical upstream bioprocessing workflow, where the goal is to maximize production of a target protein, such as an antibody, from an engineered cell line. Cell culture fermentation begins with a starter culture of cells. These are fed into various bioreactor vessels where different conditions can be used in order to try to find the best solution and best parameters for growing and optimizing your protein yield. There are many inline measurements made throughout this process in the bioreactor, such as temperature, pH, redox potential. And in addition to that, aliquots are taken from different vessels at different time points and measured using things like a cell counter, plate reader for protein titer, or metabolic analyzer. Each of these different instruments, as well as the bioreactor, generates a data output. Oftentimes these are exported manually from the instrument. The data then typically needs to be transformed, often with Excel manually, into a format that's more usable for the endpoint of the experiment. These different files need to be stored somewhere, either locally or in the cloud. Information from these experiments needs to be documented in an ELN or a LIMS system in order to describe the different experiment conditions that are being tested and the results that are being generated. And of course, oftentimes, these have to be transformed to view in an analytics tool to get greater insights into the process. Even with just one of these files, this is a time-consuming process. Never mind the fact that you have to do this with all of them. So the pain points in this current workflow are all the different manual transfers and transcription of data in order to achieve each of these endpoints. There's a lot of room for manual error as well when you're doing these transcriptions. Beyond that, there's really no integration between different functions, such as the downstream protein purification or protein characterization functions. Everything is kind of contained to the individuals who are doing this process and their own method of organization. Once these files are saved, it can still be difficult to find them because typically this relies heavily on naming conventions and file names or in folder paths that they're stored. This organization system might not be easily understood by someone else who comes into the lab. Because of all the effort that it takes to manually curate this data, there can be limited or no trending capabilities between data over time and different experiments that have been performed. And this, of course, all leads up to the challenge that this is not a scalable process. Tetra Science is going to replace all of the manual data capture that is currently happening. We can also replace all of the transformations that are taking place and the export and automatic push into the various data sources. We enable secure and centralized data storage, reduce the scientific labor in these transcriptions. We ensure data integrity by making sure there are no transcription errors in the process. And of course, this is all facilitated by automation in the platform. Beyond that, you can always go back and find your files because you can now search and query and aggregate the results in our platform based on various time points, experiment names, or even conditions that are used. All of this is made available for consumption through API and SQL endpoints. This can help with the further integration downstream. Now I'm going to show you a dashboard that we created on upstream bioprocessing data as an example of what an endpoint can look like with Tetra Science. We're looking now at a Spotfire dashboard that has been built on top of Tetra data and using the SQL endpoints from the Tetra data platform. This is a near real-time dashboard that can be refreshed within minutes of results being captured. In this dashboard, we're able to look at the results extracted from a bioreactor. We can see measurements of temperature and redox potential, as well as pH over time. We can do this for any of the sample vessels and experiments that are currently being run. In another dashboard, we're able to look at the results from the offline measurements of our cell counter, such as cell viability over time or total cell count. In the next dashboard, we're looking at protein titer results using our plate reader. This might be from our ELISA assay. Here we can look at the various samples, view them as plates in the dashboard, and we can track over time how this protein titer increases for a particular sample. This data becomes even more powerful when you start to combine it all in one dashboard. So we can look at titer versus things like pH or titer versus temperature over time. And really, it's the combined value of multiple measurements and multiple data sources all in one place so you can have a deeper understanding of what's happening in your experiment. I'm now going to quickly show you how this solution looks within Tetra Science's architecture. First, on the left, we have a number of the instruments that are used in that bioprocessing workflow. Our Amber 15 bioreactor, a ViCell cell counter, and a plate reader that would be used for protein titer. Tetra Science offers productized connectors as well as agents that can automatically collect data from these instruments. Once they collect that data, we can use pipelines in the platform to contextualize and extract data from file paths or static metadata from things like the agent configuration in order to bring that raw data into the platform, as well as attach labels to it that enable advanced search capabilities. Next, we'll engineer that data into Tetradata. So this involves remapping and restructuring raw data into a JSON format with a standard ontology, so that something like sample is always called sample, regardless of how the vendor stores that in their own file. This data is then made accessible through SQL and API endpoints to analytics dashboards. And because we've standardized ontologies, it also makes it very easy to export this for machine learning processes downstream. That's all I have for today. Thank you for joining.